The DNA double helix contains two sequences of nucleotide code letters that run along the molecule. Untwist the spiral, and DNA can be seen as two parallel strands. That's why it's sometimes called the double helix. Unzip the strands, and you have two linear sequences of the letters A, C, G, and T. The precise order of the letters along the molecule carries the coded instructions. These are the equivalent of binary ones and zeros in a computer. One strand is a complementary image of the other, and A will always pair with T, and C with G. So if you know the sequence of one strand, you can work out the sequence of the other. It dawned on Watson and Crick that this complementary copy of the code was the key to understanding how genetic information could be passed on. So the essence of life could be explained with chemistry. There were no mysterious life forces. During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present. The structure of DNA that Watson and Crick discovered suggests how genetic information is passed on. Before a cell divides, the double helix unwinds and the two strands of the DNA molecule in the nucleus separate. Each strand is then used as a template for the construction of new DNA molecules. The replication of DNA is simple in theory, but much more complicated in reality. The precise details have only recently been worked out. Take a look at the animation called Replication Mechanism to see what actually happens at the molecular level.